So there are three main types of neurons to be familiar with. Our sensory neuron carries impulses from the receptor, the part of the body that receives the stimulus, to the spinal cord. So this could be from the eye to the spinal cord, the skin to the spinal cord. Then we have our connectors, which are our association neurons, and they connect the neurons in the central nervous system. Our motor neuron carries impulses from the central nervous system to the effector, which could be a muscle or a gland. All neurons have the following things. So a cell body, which contains the nucleus, mitochondria and other organelles, the dendrons and dendrites that carry the impulse towards the cell body, our axons that carry the impulse away from the cell body. And in our three diagrams, we can see that the axon and the dendrons are different lengths in each type of neuron. Our motor neurons are the one we commonly think about, where we have short dendrons and dendrites and a very long axon. But our sensory neuron, you can see the dendrons and dendrites are longer, travelling to the cell body, and then you've got again a long axon leaving the cell body, going out away from the cell body. Axons will always terminate at a synaptic bulb, and some neurons are myelinated, which we'll look at later, meaning that they are covered in myelin sheath, which are made by the Schwann cells, and these neurons have gaps on them called the nodes of Ranvier. When a cell is at rest, when there is no stimulation of the neuron, then we have what is called the resting potential. So this is the potential difference across the cell surface membrane between the inside and the outside of the axon. This is normally sitting at minus 70 millivolts, where we have more positive charge on the outside compared to the inside of the axon. The neurons are said to be polarized because we have this electrochemical gradient across the membrane with more sodium ions on the outside compared to inside. As I said, the resting potential value is minus 70 millivolts, and this is maintained by a number of ways. Our sodium ion channels are closed, so sodium ions cannot move into the neuron. Potassium ion channels are open, so potassium ions can diffuse out of the axon. And then we have our sodium-potassium pump that is actively transporting 3 sodium ions out and 2 potassium ions in. And remember, sodium ions and potassium ions are positively charged. So we end up with more positive ions on the outside of the axon compared to the inside, giving us the resting value of minus 70 millivolts. So an action potential is whenever a neuron is stimulated. And when it is stimulated, it changes the permeability of the cell surface membrane. This causes positive sodium ions to diffuse into the axon down the electrochemical gradient. At the threshold value of minus 55 millivolts, sodium ion channels will open, and we get then rapid diffusion of the sodium ions into the axon, and this is known as depolarization. And you can see that the graph changes from minus 55, a sharp increase, to plus 40 millivolts, because we now have lots of positively charged sodium ions inside the axon. At plus 40 millivolts, the sodium ion channels then close and our potassium ion channels open, and repolarization occurs. So the positive potassium ions diffuse out of the axon, so there are now more potassium ions on the outside of the axon, and this will cause an overshoot where the inside of the axon becomes more negative than the minus 70 millivolts that it would have at rest. And this overshoot is known as hyperpolarization. And both repolarization and hyperpolarization are in this period of time known as the refractory period. And during this time, the membrane cannot be depolarized. During hyperpolarization, the sodium-potassium pump is actively transporting 3 sodium ions out and 2 potassium ions in. So again, we have lots, because of depolarization, there are lots of sodium ions on the inside, and during the refractory period and hyperpolarization, the sodium-potassium pump has to get those sodium ions out of the axon. So the importance of this refractory period is that it will ensure that the action, not cation, potentials are propagated or moved in one direction only. It will limit the number of action potentials that can be fired, and it ensures that each action potential is discrete. There is the all or nothing law, which is linked to this threshold value. So during depolarization, when the membrane becomes permeable, ions start to move in. When it reaches the threshold value of minus 55 millivolts, then an depolarization and action potential occurs. Before this, between minus 70 and minus 55 millivolts, an action potential will not be triggered. So if there is not enough movement of sodium ions into the axon to reach that minus 55 millivolts, then depolarization will not occur. It is only when it reaches minus 55 millivolts, enough sodium ions have moved in, that depolarization and an action potential occurs. Once an action potential is started, then we get the all. It will travel along the neuron as a nerve impulse. The impulse is propagated along the neuron as it will trigger the neighbouring sodium ion channels to open. So we're getting depolarization happening as our sodium ion channels open. That triggers the next sodium ion channels beside them to open, and we get depolarization moving along the neuron. So this propagation of the nerve impulse, the movement of it along the neuron, is speeded up in a number of ways. So the main way is having a myelin sheath. So the myelin sheath made by the Schwann cells acts as an insulator. There are tiny gaps known as the nodes of Ranvier along the myelin sheath where there is no myelin sheath present, and it is at those nodes of Ranvier that depolarization can occur. So we say that the impulse jumps along the axon, and this is known as saltatory conduction, and it's jumping from one node of Ranvier to the next node of Ranvier. And this will speed up the movement along the axon. Other ways it can be speeded up: the diameter of the axon. So a thicker axon will cause a faster impulse as there is less leakage of ions in a neuron that has a larger diameter. Ones who have a smaller diameter, there's too much leakage of ions, which makes it much more difficult to maintain the potential gradients across the membrane. Higher temperature will increase the rate of diffusion of ions and therefore increase the rate that depolarization could occur. At the end of the neuron, there is the synapse. So we have the presynaptic neuron, the postsynaptic neuron, the synaptic cleft. When an action potential arrives at the presynaptic membrane, it will cause calcium ion channels to open and calcium ions diffuse into the presynaptic neuron. This in turn then causes presynaptic vesicles to move and fuse with the membrane and release the neurotransmitter, and in this instance we're thinking of acetylcholine, which is ACH here. The acetylcholine diffuses across the synaptic cleft and binds to receptor proteins on the postsynaptic membrane. This will then cause the sodium ion channels to open in the postsynaptic neuron, and again, once minus 55 millivolts has been reached, the threshold value, then we're getting that all or nothing response where depolarization occurs and our action potential propagates along the postsynaptic neuron. So this is an example of an excitatory synapse, which will cause an EPSP, excitatory postsynaptic potential, which can lead then to an action potential. So an EPSP is the potential change in the positive direction due to the opening of the sodium ion channels in the postsynaptic neuron, i.e. changing from minus 70 millivolts to reaching that minus 55 millivolts. So it is capable of generating an action potential if we get to that threshold level. If the threshold value is reached, then depolarization occurs and an action potential is produced. Once we've had our action potentials, then acetylcholinesterase, an enzyme that's found in the postsynaptic neuron, will break down the acetylcholine or hydrolyze the acetylcholine to ethanoic acid and choline. This is important so that the sodium ion channels do not remain open and action potentials are not then being continuously generated in the postsynaptic neuron. The ethanoic acid and the choline then diffuse back across the synaptic cleft and are reabsorbed into the synaptic bulb of the presynaptic neuron, where they then are resynthesized to acetylcholine and stored in the synaptic vesicles to be used again. And this is where the mitochondria play their role, where they release the ATP that's required for the synthesis of the acetylcholine. So the advantages of having the synapse It allows nervous communication to continue throughout the body as impulses can travel from one neuron to the next. It ensures unidirectionality. So the action potential can only pass from the pre to the postsynaptic neuron, as neurotransmitters are only made in the presynaptic neuron and the receptors are only found in the postsynaptic neuron. It prevents overstimulation of effectors. If there was too many impulses, that would quickly, then the supply of the neurotransmitter would be used up and the synapse would fatigue. And it provides integration, which is what the diagram is showing you here. A number of presynaptic neurons can synapse with one postsynaptic neuron, which will then provide flexibility. So getting information, if you think of it, coming from lots of different places, then being sent along with that postsynaptic neuron. An inhibitory synapse is one that makes it more difficult for that excitatory postsynaptic potential to be generated, i.e. harder to reach the minus 55 millivolts. This is because chloride ions enter the postsynaptic membrane, which lowers the potential difference to below minus 70 millivolts, which is similar to our hyperpolarization in the diagram there of an action potential. And this decrease is known as an inhibitory postsynaptic potential. So it may be a term that you might see coming up in an exam paper. So as I said, it makes it more difficult to reach the threshold value, so therefore more difficult to create an action potential. Now, why there are inhibitory synapses is that it reduces the input from background stimulus that would clutter up the nervous activity of the brain. Summation. There are two types of summation to be familiar with: temporal and spatial summation. So summation is the sum of the neurotransmitter that will add up together to help an action potential be achieved. Again, reaching a threshold value that will give the all or nothing response. Spatial summation, which is what the diagram on the right is showing, is defined as signals coming from multiple simultaneous inputs, i.e. more than one presynaptic neuron. As you can see in the diagram, you can see that if A, B or C are just releasing neurotransmitter, there is not enough being released to cause an action potential. However, if all three are fired simultaneously, then the sum of the neurotransmitter being released to the postsynaptic neuron is enough to trigger an action potential. Temporal summation comes from repeated inputs. So again, if you look at the diagram on the left, two firings with a pause between them causes no action potential. There's not enough neurotransmitter from each firing to reach the threshold value. Three firings in rapid succession, however, cause the neuron to reach the threshold and an action potential is triggered. So essentially, to achieve an action potential, the threshold voltage must be reached at minus 55 millivolts. This can be determined by either adding up the individual inputs from temporal and spatial summation. There are two main types of neurotransmitters to be familiar with. And remember that each synaptic bulb will have its own type of neurotransmitter, only produce one type of neurotransmitter. So acetylcholine is the main neurotransmitter of our excitatory synapses found in the central nervous system. Another one is noradrenaline, which is involved in the involuntary control, like gut movement. And then an example of an inhibitory synapse neurotransmitter is GABA, and it is released, and its function is to help reduce anxiety and panic attacks. Drugs can also stimulate or be antagonistic to neurotransmitters. So nicotine stimulates the release of acetylcholine and other neurotransmitters, so action potentials are more likely. And that's just a diagram showing you the nicotine mimics the action of the neurotransmitter acetylcholine, so therefore more sodium ion channels open, more action potentials can occur in the postsynaptic neuron. Curare blocks receptors at the neurotransmitter junction, so prevents synaptic transmission, and this would result in a loss of muscle function. And the bottom diagram is just trying to show that, where the curare binds with the receptors that would normally bind with the acetylcholine. So if the curare is bound to it, less acetylcholine can attach, therefore less sodium ion channels can open. And then opioids, they will block the calcium channels in the presynaptic neurons, so less transmitter substance can be released, Therefore, action potentials are less likely to happen. And this will be used for pain relief because it reduces impulses to the brain receptors.